All right, good morning, everyone, and welcome to the Introduction to Single Cell Analysis tutorial. My name is Sarah Monroe, and I am here today with my colleagues, Natalia Calixto-Mancipi and Alyssa Williams, and we will be giving instruction today. Natalia will be the co-instructor, and Alyssa will be moderating the chat for us. So this is the tutorial webpage that we have. All of our tutorial materials are here. This is a newer format for us, so you are the guinea pig class for this. We decided to move this from a purely lecture-based format into both lecture and a bit of a demonstration, with the goal of moving this into an interactive hands-on tutorial for you in the future. So you will be able to see the recording of this tutorial on YouTube after it's completed. And if you ever have questions, you can reach out to MSI here at these links. You can contact help@msi.umn.edu for general help. And then if you want to get in touch with the bioinformatics team specifically, you can email us at ribhelp@msi.umn.edu. So to start with, we want to talk about what we're going to cover in this tutorial. So we have several goals today. First, we're going to discuss single cell measurement technologies, including some guidelines for planning a successful single cell or single nuclei experiment. We'll provide a broad overview of the gene expression analysis steps for these types of experiments. Natalia will give a demonstration of how to use MSI's resources for analysis in R and Python. And we'll discuss some important analysis considerations. And then we'll showcase some possible analysis avenues for your annotated data, and we'll discuss some other data types. One point I want to make here is the most of the time in this tutorial, I will talk about single cell, just because it's easier to say single cell rather than single cell or single nuclei. But both of these workflows are very similar. There's a couple of minor differences in terms of sample preparation and quality control steps. And we have some details on that and can get into that later during the question session. So to start with, we'll go through and talk about why we are interested in using single cell methods. So your tissues are heterogeneous, or you're looking at mixtures of cell populations, and you were previously only able to use bulk RNA-seq, which allowed you to measure the averages of all cells in your sample. And single-cell RNA-seq allows you to actually reveal specific developmental patterns or subtle differences in cell type. It's become really a powerhouse for immunology studies and also cancer biology studies. And so here we're showing this cartoon to describe the differences between the workflows. Essentially, you have this heterogeneous tissue for bulk RNA sequencing. It's essentially becoming a smoothie of your given sample, and you end up with gene counts for a given sample. Whereas with your single cell technology, you're going to get a matrix where you have your gene counts for individual cells in your sample. And so you're really going to be able to profile your individual variation. So to give you a little bit of a context for where we've been with single cell sequencing, it's grown tremendously over a decade or more. And so this is a nice chart demonstrating how things have scaled. This is actually a log scale here in terms of the number of single cells in a given study. And so these were the early days of academic lab-based technologies. One of the first platforms that people used that was widely accessible commercially was the Fluidigm C1, where you could measure 96 cells. So we're right here around 100 cells. And then today and beyond, so this is from a 2018 paper, we have 10x Genomics and SplitSeq, which is also known as Parse Biosciences. And these are kind of the dominant technologies right now where we're actually beyond 1 million cells. We can sequence close to that number, and they're really pushing the limits even further. And so when you think about the scale of this, you need stronger compute power. It can be hard to analyze these data sets, or actually impossible to analyze these data sets on your laptop. And so that's why we have the supercomputer. So we'll just talk briefly about these two major technologies, the 10x Genomics platform and the Parse platform, because those are what most researchers are using right now, although that could change in the future. So with the 10x platform, you're relying on microfluidic cell encapsulation in droplets. And so these are special beads that are coated with DNA molecules that are called gems that are encapsulated with the cells. And then those are collected. And on the bead surface, there are DNA molecules that can be used to label all the individual transcripts from a single cell. So you have both a cell barcode and a UMI for a transcript barcode. And this has been a really powerful technology, and a lot of people are using it today. Combinatorial barcoding is a relatively newer technology that researchers are using here at U of M. And the kit that most people are using is the Parse Evercode kit. And the idea here is that you're utilizing combinatorial barcoding rather than microfluidics. So you're essentially taking your sample of your cells and pipetting a single sample into a 96 well plate well. And then you, at that initial step, you're doing reverse transcription, and then you pool all of your wells together and then redistribute them into a second plate where another barcode is added, and so on. And you continue through this process until you have 4 different barcodes. And then those barcodes are used to distinguish a given gene within a given cell. So that's a brief background on the type of data that we're analyzing. And now we'll get into the some of the design considerations for your experiments. And so depending on your biological system and your research questions, you have a lot of choices to make in terms of how you're setting up your experiment. We have developed, and I say we collectively, the 17 bioinformatics analysts at MSI have developed a single cell experimental design template to help you to think about what you need to do for your sample preparation and sequencing. And so I'll just click out into this link for a second to show you it. This is the MSI single cell guide page. And then there is this downloadable template document here that you can look through to give yourself some ideas of what to think about for your experiments. And there's also a Google form that you could submit, and then we can get in touch with you and talk about it. And we're happy to meet with you to design your experiments. And so some of the key questions that come out of that document include, what organism are you looking at? How are you going to avoid batch effects in your experiment? What is the morphology of the cells that you're interested in? Can you actually capture them? So neurons are tricky to capture in microfluidics. Some cells are multinucleated, so like muscle cells, and so that can be challenging too. And so you might want to consider different approaches rather than a droplet-based approach. And then also you might need to do optimization of your protocol for actually isolating your single cells because of stickiness and tricky dissociation issues. And then another thing to think about is how abundant the cells that you're interested in are in your tissue. You may want to do some sort of sorting or enrichment. And then, of course, one really important point is that if you're planning to do statistical analysis, such as differential gene expression, then we strongly recommend biological replication. So now I'll hand it off to Natalia, and she will discuss the workflow overview.
Hello, my name is Natalia Calixto, and I will be helping to deliver the rest of part of the tutorial. I am also a core analyst, and you can contact us, as Sarah said before, for any questions that you might have from the tutorial or other things. Okay, so let's imagine that you already designed very carefully your experiment, and now you're ready to collect your samples. So you will collect your samples and dissociate the cells, and then you are going to need some library preparation and sequencing. These steps are usually done by a facility like the UMGC. So when they are done, they deliver you something that's called the sequencing files. And that's when you start with the analysis. The analysis is long and it has many intricacies. So the goal of this part of the tutorial is just gives you like an overview of what you need to do and why you do it. The first step in this path is processing these sequencing files, right? So they are usually in a format that's called FASTQ, and we'll go a little bit into it later. But then when you process these FASTQ files, you want to end with something numerical that you can analyze more. And this is the gene counts matrix. So this is step 1. Then you go to step 2. Step 2 starts with the counts matrix, which you further analyze more and more, like do statistical stuff to it, so that you can extract the information of the cells, of the cells that are in your sample. And then you have to maneuver with the data a lot so that you end up with a data set that makes sense biologically, which we call annotated data set. We'll go more into details further as we go through the tutorial. But bear with me. Let's imagine that you got your annotated data set. Then you go to step number 3. And this is where you actually start to answer or explore the biological questions of your experiment. Okay, so let's start with step 1, a little overview. What is this sequencing file thing? Like, what are you talking about? Maybe some of us are not familiar with this. So turns out that a sequencing file that comes in a FASTQ format is basically a really big text file. And this text file has information of every single sequence that you got in your library. And it looks like this. So for each sequence, you have a row, a line that is a header. It's like a name tag for each read. And then you have the sequence itself, ATCG, whatever. And then a plus, and then a quality score. So your goal in this first step is to compare each one of these sequences, that could be millions or billions or more, to the genome of your organism, the organism that you're studying, and count how many times you find a sequence that resembles a gene in that genome. Then tally all these numbers by gene and by cell, and then do some QC with those numbers. That is the alignment part, is the comparison. And then the quantification and filtering is the counting and tallying and QCing. But although it sounds kind of onerous, don't worry, because we have standardized this process from like it's been there for a while. So we have pipelines that do the job for you automatically. You have Cell Ranger if you're working with 10x Genomics, and you have a split pipe if you're working with parse. And really, the only thing that you need to do is write a script that can look like this to send the job to the supercomputer. So you would make a file in which you tell the supercomputer, hey, I'm talking to you in Bash. And then you ask for some resources like memory and how much time you need it. Then you tell the computer, hey, I'm going to use this software, which either it's installed in the system, so you would module load it, or you could tell it, I installed it myself, and here's where to find it. And then you put the commands and the options that are particular of your sample. If you're going to do this yourself, what I recommend is that first, you check the documentation of the software that you're going to use. This is a link for the Cell Ranger documentation, but SplitPipe also has some documentation on how to use it. Also check if we already have installed the software with the module avail command so that you don't have to install it. And also check, because sometimes, I mean, if you're working with mouse or human, and other organisms as well, we have pre-built the genome references that you need to compare to. And we host them in the common bioref folders here. So check if we have that so that you don't have to repeat work that we've already done for you. And then, okay, and then you just finish your script and send it to the scheduler with the sbatch command. And then if it runs completely and it's successful, then you should receive an email, or if it doesn't, you should receive an email that tells you about like what resources were actually used, I recommend you look at this email so that you can troubleshoot if your job failed, or also fine-tune the resources that you need for the next time. Okay, so that was part of it. Now we move to step 2. And here I'm just gonna make a little stop and ask you to, if you have questions, please raise your hand or interrupt me or something throughout the tutorial. It's good. All right. So step 2, we have a count matrix, which we just generated, and we want to get to an annotated dataset. The count matrix is basically a humongous table in which we have a number for each cell and each gene. That's what it is. So we start by making some quality control, because if you're using, for example, droplet-based methods, maybe you are reading things from a droplet that's empty. You don't want to do that in your analysis. So the initial part is a quality control. But then you also have to kind of modify and wrangle this data because it's humongous. You have thousands and thousands of cells, or millions, and then thousands and thousands of genes. So you have to do some normalization and dimensionality reduction to be able to make this data more manageable for computing. And once you do that, then you go to another part that is the cell clustering and the annotation. And this, in other words, is basically seeing which cells are similar based on the numbers in this table, which are the expression profiles. Those are the clusters. And when you have your clusters, then you want to try to know what they represent, what cell type they are. And that is the annotation part. This is a tricky part of the analysis, and we'll get into it later on. Okay, and once you have your annotated data set, then you can start asking questions, like, for example, is the treatment X changing the gene expression in my cells? So that would be a differential expression analysis. But you can, that's very typical, but you can also ask other questions like what genes seem to be working together. That would be a network analysis. Or maybe your treatment is changing the proportion of cells in the samples. That is another question. Or maybe you're interested in some differentiation path, and that would be a temporal modeling question. There are so many questions that we really can't go through all of them, but this is an overview. Okay. Any questions so far? Okay, good. So is good enough theory, like enough theory for now. So let's do some demo. And in the guide, you have the slides that have screenshots of what I'm going to demo, but we'll just get right on. All right. The first part that I want to show is Open on Demand, which I hope we are familiar with, but it's something that is super useful and some of us might not be. So let's try it. We just go to openondemand.msi.umn.edu. And right here. That brings us to a landing page. Well, first, you will have to log in, which I've already done, but you will have to log in and then authenticate with Duo. This is a super great resource that we have because it allows users to browse and connect to the supercomputing resources, even if we don't feel so comfortable with the command line and connecting through a command line. So you can use the Files tab. If you click here, you will see all the projects that you belong to, and you can click in any of these directories, for example, your home. These files are stored in the basement, right? But we can look at them just through here. So the Files tab lets you browse through the documents that you have in different folders, and you can preview the files as well, depending on the size and the type. But for example, this is a little text file that I have. I can just click on it and I can preview it, or I could even edit it here. And also you can upload and download things, and that can be very useful. Okay. The second tab that I want to show you is the Clusters tab. And if you click on the Agate Shell Access, then This is a very easy way to access the supercomputer in an interactive manner, but using a terminal. So here we have a terminal and we can talk directly with the supercomputer. We are within the Agate node. So I can tell it like, tell me where I am, print working directory. You are in your home directory. Or tell me what is in my home directory, right? Right. Or I can tell it, take me somewhere else, like change directory, scratch global, value 015. That's my directory in scratch global, right? Perfect. So this is a way that maybe it's easier to start with in the terminal. And finally, I wanted to show the interactive tabs app because we'll go into this later, but basically it's just a way that you can use the supercomputing resources, but with apps that you normally use in your laptop, like RStudio or Jupyter, or maybe IDL, Mathematica, whatever. Just click on them and then you ask for resources in the supercomputer. Okay, so that's what I wanted to say here. Any questions? Okay, so now that we've learned how to access Open on Demand and how to do things in the terminal in Open on Demand, let's use that knowledge to do some single cell things. And I think this part is going to be really useful for people that are hoping to learn how to analyze data, but they don't have data yet. So they can just get some data from a public repository like Geo. So I have done some browsing and I have the data, the accession number of a data set that I want to look at in Geo. So I'm going to copy it and go to the Geo website, which is this one, NCBI. Well, you can just Google Geo and it'll take you here. And here in the keyword or accession thing, you can paste the accession and click search. Boom. That leads you to a landing page that shows you information about the sample that you are looking or the data or the keyword or whatever. You can just browse here. I've selected this one because it's a little data set that has single cell RNA-seq data from 10x Genomics. And because if you scroll down, the submitter was very kind and also added the feature barcode matrix. In other words, they added the counts matrix, which is what we want to download. So to download it, you could just click on the FTP and that would bring it to your computer. And then from your computer, you can upload it to the supercomputer. But that's cumbersome and it's also very slow if you're working with big data sets. So I want to show you how to do it on the command line. If you just hover here, you'll see on the bottom of the page that this link is trying to get you to somewhere. That's the FTP site of NCBI, Geo, and then the samples folder, and then some other folder, something. That's a long path in the bottom of the screen. Do you see that? Mm-hmm. Like here, here, there. Yay. Don't worry if you don't see it. I have made a screenshot for you, which is in the web page. It's trying to take you here. Okay, so this is basically where that data lives. Okay, so it's going to be in the FTP part. So let's go to home in Geo and go to the FTP site. Let's go to the samples, which is, we're following this FTP in Geo, the samples, and then we're going to go to the GSM8 for whatever. It's a big, big list. It's a long list because it's all the samples that are in Geo. But anyway, once this ends loading, we'll just look for this folder and then continue this one. Right. So let's look for what was the number? 8475. 8475. here is another folder. Click there. And then we're going to look for 8475.184. Right, now 184. This is the folder that contains our data that we just saw in here. If we go to the supplementary, then we find the feature barcode matrix that we want to download. So let's go to our terminal. In my Scratch Global, I'm going to make a directory that's going to be a single cell intro. Change directory single cell intro. And then I'm going to be fancy and make a directory that's going to be called data geo. Oh, sorry. Cool. Go to data geo. And here I'm going to use a command that's called wget. Wget is able to connect to a URL and download something. So this is the URL. We just copy and paste it here. And this is the something that we want to download. We just copy and paste it here and hit enter. And that's it. Then we do list and we have our data in the supercomputer. Questions?
I have a comment that
Yes.
I'll let users know. So Scratch Global is a really good place to work, especially if you're downloading large data sets, because your user home directory only has 200 gigs of storage. You don't have very much space. Or you could also work in your project space. And Natalia has her own directory in Scratch Global that she had to make. And so you would have to make your own directory in Scratch Global. It's not something that MSI makes for you.
Okay. So in the tutorial guide, I have literally, I have just written the commands. So if you want to try this later, you can copy the commands and make the things. The only change is that instead of like seeing Calix015, which is my user, you would see yours. But you can try this later on and see if it works for you. Okay, if you were working with a UMGC, then they would deliver the data directly to your MSI space, which can be found in the data delivery UMGC folder. So you wouldn't have to download anything, it's there. Okay. That was how to get data. But we haven't started like working with it. So I wanted to help introduce you to the two main packages that are used for analysis of single cell data. They are ScanPy, which is Python-based, and Surround, which is R-based. And we can use them through the Open on Demand interface. So if you go to the interactive apps and click, for example, Jupyter, then you can go and select the options that you want to use for the supercomputer, the resources. Usually I use lab and this is fine. And then the group that you work with, how many resources, and whatever you want to do, the time, and then you click launch. That will take you to the interactive apps page where you would see a Jupyter session that is going to be in a queue state. I did this a little bit before so that I wouldn't have to wait. But once you're here, then you can just connect to Jupyter. And that's it. You are working with the supercomputing resources, but in an interface that might be familiar to you, which is Jupyter Notebooks. You should know that I have pre-installed the software that we need for this tutorial. But when you're running your own analysis, you will have to do some installations. That can be tricky. So I have put some tips in the tutorial guides. But I really recommend that you check out our YouTube video of software management that another staff member gives, and it's really good. And also, if you have questions or issues, just email us and we'll try to help. Anyway, I have installed a kernel, basically a bunch of software for this tutorial. So I can just click here, boom. And we see that this appears. It has, this is the notebook interface, and there are chunks. This is called a chunk where you can put either code or plain text. And you can also look at different files here. So we are going to do something with it a little bit. Okay, if you want to rename this, you would go right click and rename notebook, single cell tutorial, or here, whatever, rename. And you see that it appears here. It's in your files, in your home directory or root directory. And so the first thing that we want to do is tell the notebook where is the software that we need to use. And we do that with the command import and scanpy. To run whatever is written in a chunk, in notebooks, you would press Shift and Enter. So then the computer is like, oh, I'm looking for scanpy. And that's it. I'm gonna look for that. While it runs, you know it's running because it's in a star, and then when it finishes, it just gives you a number, which is the order of operations. Okay, so now I want to tell it where my data is gonna be. So I tell it I'm gonna make a variable that's gonna be in data that will store what the geo data that we just downloaded is. So let's start with, I know it's in Scratch, dot global and calyx 015, and then you can use the tab to complete. And I have single cell intro, tab, data, tab, geo, tab. If you just click tab, you don't have to type so much and it's easier. All right, so that's my variable, what the data is. And then I want to use this data to make something called an AND data object. An AND data object is what scanpy uses to store single cell data and to analyze it. So for that, I'm gonna use the function within scanpy. So scanpy, something within scanpy, the function that's called read 10x h5. Parentheses, my data. Shift enter. Okay. Boom, it happened, but it gives me a warning. What's going on? It says the variable names are not unique. To make them unique, use this. All right. But for now, I don't know what you're talking about because I don't know what is AND data. So let's say we're gonna look into the object. And here, I just want this to be plain text, so I do like this, and it's just plain text. Okay, so if we just type a data, then it tells us that is an object with a big, big, big number here, plus times a big number here. Okay, but it's something called an AND data object. So I have borrowed this figure from those scanpy people, and you can observe it here. It's an, well, I don't know how to make this smaller. You know how to make this smaller?
Maybe just, yeah.
Ah, okay. So it's a scheme of the AND data object. Basically, everything goes around this green thing that's an X. Spoiler alert, this is the expression data in the counts matrix. And you can see that it has several layers, so we can do different things to the expression data. And everything goes around the axis of this counts matrix. Either in this axis, we have the var, and in this axis, we have ops, and then we have more things. So because of time, I will only use the var and the ops. I will only talk about these three things, but know that there are more things. And if you are interested, you can just look into the AND data tutorials, which I have also linked in the single cell guide. All right, so all right, we saw a bunch of things, and then we saw that there's an X, and there's vars, and there's ops. What is all this? So we can just ask with type a data ops or var, whatever you var, because it gave us an warning. What's this? Oh, look, it's a data frame, a pandas data frame. That's cool, because a data frame is basically a table with names, like with index names. So we can do like add data dot var dot head, and we can look into it. It's a table. It's a table, and we see that the row names of the table are the genes. And obviously, it's telling you like, hey, those names are weird, are not unique. So if they are not unique, there are duplicates, right? Okay, so let's see if there are duplicates. You can access the names like add data, var, tab, var names. What is this? The gene names. And okay, so let's say, how many are they duplicated? No. Okay, let's count. How many are true? Okay, so count them. We have 95 duplicated names. I'm doing this so that you kind of see that you can access different things within the data object with the dot operator. So for accessing the variable names, you do dot. And to see if any of these things are duplicated, you dot duplicated. And then, since this is a vector, you can also add whatever is true and sum it, right? Okay, but the next important thing is to read the warnings and pay attention to what they say. These are telling you the variable names are not unique. Yes, we saw that. And then to make them unique, call var names make unique. Perfect. So we're going to do a data var names make unique. You see that I'm using the tab as much as I can because it just makes things very easy. And now we're going to run this again to see if it actually made them unique. And it did. There are no duplicated anymore. Okay. So those are the gene names, and the information about the gene. But we also have cells in our data set. And this is stored in the observation, in the observation slot or ops. All right, so the ops, it's also type, let's see what it is, add data, ops. Hey, look, it's also a data frame. So ops, head, let's look into it. And we see that it only has the barcodes, which by definition are unique, so we won't have the problem of the names being duplicated. All right, so we can add more things to the ops data frame. We can, for example, say, I want to add more information like that they come from this geo-accession. So you type that, hit enter, and then if you look at the head again, you see that it's added, right? So at the end, while you do your analysis, you'll be adding a lot of columns to this data frame. And that is where the analysis results are stored. Okay, finally, and very quickly, I want to look at the expression data, which is stored in the X. So if we look at X, it tells you that it is a sparse matrix, that it has these dimensions, which is, this is a lot, it's like 1.7 million times 32,000, but it only stores about 19 million elements. If we do some quick math here, and you multiply this by this, actually, it gives you, I mean, we can do it. It gives you a really big number, which is much bigger than this one. So what's going on is that in a sparse matrix, you only store the values that are non-zero. That means that in our sequencing count matrix, most of the values are zero. So if that's true, let's check it out. This is X, and we want to see how it looks. And you see that most of them are zeros, and this is just a subset of the thing. So this is what we mean when we say that single cell data is very high dimensional, because it's a humongous matrix, and it's sparse because most of the slots are zero. Okay, so this actually needs to be dealt with because computing with this sort of data it's silly because most of it is zeros and humongous. So what we're going to do is modify the expression values in this X. But to do it safely, you could say, I want to copy this data in X to another layer, which is the things that were stacked under X, so that I have like a copy, like a back, I don't know, something that I can go back to. So to do that, I will do add data dot layers. So I am accessing the layers part of data, and I'm going to create something new that's going to be called counts. I'm going to create something new in the layers. And I assign what I have right now in X. And I want to make a copy of it. That's it. So now, if I just look into what is data, then you see that it's an object, humongous, that has the geo column in the ops, which we just added, and that has the variables, which are the information about the genes. And we also added a new layer, that is the counts. So basically, like, we could go further into this, but we don't have the time. So I would encourage you to, based on these little things, try to play and go into the tutorials and see what else you can find. But the last thing that I wanted to show you is how to save your work. Because you could do a ton of calculations and modifications, and then you just exit. You just lost all your work if you don't save it to disk. And the way to do that is add data dot write. And then you wanna put the path where you wanna save it. Like if I wanted to do it in my home directory, I would just do the file name there because that's where I'm currently at here. But I want to do it in the Scratch, which is where I put my the counts matrix. Single cell tutorial, and then that data dot H5A. That's the common format that is used to save it. And if you do that, then it's saved there, and let's check it out. Change directory. Is it here? Nope. We just save it here, and now we can access it later on and continue our analysis. Okay? So that's all I wanted to do for ScanPy. If you have questions, please ask. But in the meantime, we wanted also to give a little break so that people can get a breather, and then we'll come back. We'll come back in 10 minutes.
Sounds good.
Okay. Okay. But we'll be around if you have any questions. Okay, welcome back. We have had a little break, but now we are ready to try to see how to use ROT, which is the R-based package that is commonly used for single-cell analysis. We can use Seurat in the RStudio interface that is offered on Open On Demand. So, as usual, we go to our Open On Demand, Interactive Apps, and click on RStudio Server. Here, you can select the different resources that you ask for the supercomputer. For this tutorial, because we have a little data set, I am using only like 2 cores. And something important here is that when you install your software, our packages are known to have lots of dependencies. So you can actually tell the supercomputer where this software lives that is used for your packages in this box here. So you can add like modules that we have or whatever you want, right? Then you will customize your environment, you will tell it how many resources you want, and then you can hit launch. If you have found a combination of the custom environment and the packages that works for you, then you can save those settings and name them and then just relaunch it. Anyway, I've already launched my job so that we didn't have to wait until it was queued for the queue time. And I can find my active jobs here. Right, so you see that I have an RStudio session, which is running. Once you have that, you can just click and connect to RStudio. While this is loading, which I hope works, because the system was having some hiccups, I wanted to make a note that I have pre-installed the software that we need, all the packages that we need for this demonstration. But when you do the analysis, you will have to do these installations yourself. There are many packages that are particular to your needs. And for doing that, you have to have a folder where you're going to install everything that you have right permissions. So we'll go through that in a second. But first, the interface. Here is my interface. In RStudio, you have a space for the scripts or files that you want to watch, that you want to see. Let's look at, for example, the colors files that I showed you before. You can just click. This is a file browser space that you can just click and look. And here's where your files are going to appear. And then here's the console, which you can interact directly. You can say I equals 2. And then when you do that, then in this part, you see that you can see the variables and the values that they have. Okay. So I want to see the files that I have in my Scratch global directory, which is what I just created and downloaded the geo. So I'm going to click here and write the path. Scratch.global.helix050. Enter. And it takes you directly to that. And then you can just keep browsing as usual. All right. You can also create a blank file. And this can be any of these sorts of files. But for now, I'm going to do an R script that's going to be called single cell tutorial as well. Okay. Right. So for this part, as I told you, you will need to install the packages. And the way that the R knows where to look for packages is because it has a variable that's called libpaths. Libpaths. So if you see what's in this variable, you see where the program is going to find, or R is going to look for packages. In R, what you use for executing a line is Command Enter, not Shift Enter. So that's different. All right, so if you look into libpaths, you see that this is like where it's going to look for things, but actually I installed my packages in another directory. So what I'm going to do is modify this libpaths directory. And to do that, I am just going to make a vector, which is the C, that includes where I have installed the things, which I have the path right here in my cheat sheet. So I'm going to write it there. And then in the vector, I'm going to do comma. And then I am going to concatenate the current things that it's using for looking for software, which is these two. Okay, Command Enter. And now I have modified this variable. Let's see what it has. If you go the up arrow, you can see what you did before in the console. And now this libpaths has a new directory, which is where I have installed my things. Good. So we're good to go because R now knows where to look for things. So for single cell, we can tell them for single cell and for everything, we need to load the libraries, the same as import in ScanPy. Here we use the command library and then the name of the library. I like to use something that's called tidyverse, which is very good. It's very good for using with big tables. And then I really like a package that's called ggplot, which is for plotting. It's amazing. And then actually the actual Seurat package, which is what we use for the analysis. So when you hit Command Enter here, you can see in the console what's going on, like live. Okay, so similar to what we did before, I want to make a variable that's going to have, and you can assign with whatever you want, that's going to have the address of where my data is. So scratch global, Alex 015, and you see that I keep using the tab for completion because it's actually really good. And then single cell tutorial, also intro, and then data, geo. That's it. So that is my counts matrix from geo. The path to it. I store it, and then I want to create something called a Seurat object. So a Seurat object can be created with the function create Seurat object, right? How do I know this? Because I've gone through the documentation. And you can go through the documentation as well. If you go to the tutorial guide, I have linked for both Seurat and for ScanPy tutorials, a tutorial like links to the vignettes on how to do the tutorials, like basic things, and to their function reference. So if you don't know how to use a function, you can just go through and see, oh, the purposes in the step, how do I filter cells? This is how the function works, right? Same for Seurat. You have a link for the tutorials page. I would recommend going with the getting started, the little one, just to get started, and to the functions page, where you can just click and it gives you more. If you don't want to do that, You can also use the help that is installed in the packages with this sign, the question mark, followed by the name of the function. Create Seurat object. And then you do command enter. And here you can see the help that is available. So let's see. Create a Seurat object. So it tells you what to do. You add the cams, and it also shows you what are the default values for some of the options. So it will call the assay RNA and so on. If you use it, there is also another option that's called mean cells and mean features, and they are all both defaulted to 0. Okay, anyway. So I want to create my Seurat object with these counts data. So I say counts equals oh, sorry, no, we have to read the data first, right? We have to make a counts matrix because here I only stored the path. So a counts matrix is the read 10x H5 and in data, which is the path. You read whatever is there, which is the data from Geo, and store it in a variable code called counts matrix, which we can just look at here in the console. And it looks like this. Basically a bunch of genes, and the columns are a bunch of barcodes and then a bunch of dots that, as we saw in ScanPy, they represent zeros. Mainly, it's a big, big, big matrix of zeros. We can ask of the type, what type of data this is, with the is command. Similar to type in ScanPy, and it tells you it's a sparse matrix. Okay. So now that we have the counts matrix loaded, we can create the Seurat object telling Seurat that the count is the count matrix. And that's it. That's the minimum. It also gives you a warning. Hey, the features, the feature names or genes have underscores. We don't want that. But it takes care of it itself. You don't have to tell it, make them unique or anything. It just takes care of making the gene names unique. All right. So while this happens, that was fast. We can look at what is this thing that we just created. So we write it and see it is an object of class Seurat, which has these dimensions, 32,000 features. They call genes features in this case, and like 1.7 million samples with one assay, and we have one layer present that is the counts. We can access things inside the Seurat object as well, but we don't use the dot as we did in ScanPy, we use the @ So for example, we can say, hey, what is the active assay in this Seurat object? And we click enter, and it says, we do enter, and it says RNA. So we use the @ for that. Okay, so now here's our Seurat objects. If we want to see the gene names, we just do row names, Seurat, and we can pipe it ahead so that it doesn't tell us everything. Those are the genes, the first genes, but basically these are the gene names. If we want to do the information, if we want to see the information about the cells, then that is stored in the metadata slot. So if we access it, it's a big thing. It is, yeah, let's just look at the head. Okay, it has barcodes and it has some information here. when you create the Seurat object, it automatically shows you the total counts that each barcode has and the total genes or features which each barcode has. And you can see that here we have, for example, a barcode that has nothing. It's empty. We don't want that. And this is the sort of things that we want to get rid when we're doing the QC. But for things that are so obviously wrong, then we can also use the functions in the create Seurat object. So here we just do comma and a tab. Then you see that we can use the min cells option, which is tells you what's the minimum number of cells that have to express or show a gene so that we keep this gene. And I'm gonna do a very little, like tiny thresholds so that we don't get rid of much of the data except the things that are obviously wrong. And then we also have the mean features, and I want to say, show me at least 10 genes. So we are going to override the Seurat object, but with these constraints. It goes again. And if we look at it, then it says that instead of 1.7 million samples, we actually only have 84,000, meaning that we have tons and tons of empty things. Okay, cool. So we know where the names of the genes are. We know where the information of the droplets or the barcodes are, and we know now how to create the Seurat object. We're going to go through the counts. But I want to show you how a fully analyzed object looks like. So first, let's say that you wanted to save your work. So you could use the save RDS option command, and then you would tell it, save my Seurat object that I just created. And the file, again, we can write it here, scratch global, calyx, 015, and then single slot intro, just there, and Seurat object.rds. That is the command that you use to save an object in R. And that's it. And that way, let's look again. Let's saving it. But let's look again here. So we have the thing and the RDS as well. Okay. Okay. So this is like, there are so many other slots. And if you want to explore, you can also click here in the variables. If you click, you have a view option. And then you see that you have the metadata, you have the assays with the RNA, which is the counts. You will have other assays and other layers later. And I'm going to show you next that. So how does a Seurat object looks after you've done analysis. So I'm going to read the Seurat object that I have analyzed before. So read RDS, and that I have stored in actually it's in projects standard is generics. Kind of slow.
Shared. Oh, no, maybe.
And I have single cell tutorial, and then I have the data, Turkey, epithelial, so I can read it. Okay, so that's why the tab is very good, or because the paths sometimes are long. Right, so this is a teeny tiny object that I took. It has only 3,000 cells and 16,000 genes, but you see that it has many more stuff. We have something called variable genes. We have 3 layers, not only the counts, which is the raw counts, what we downloaded from Geo, but also we have data and scale data. And we also have something called dimensional reductions. You have many options here because I tried different things with this data set, but this is what you use to make the plots that you put in the papers. So let's see how the gene expression is modified with the analysis. If you look at the RNA assay and then the counts, you see the matrix that we're familiar with, but lots of dots and then integers, like this is 1 count, 2 counts, that's it. If you look at, let's copy this, the data, now you also have many dots, but you don't have integers because this data has been normalized. You have decimals. And then if you look at the scale data, we cannot even look at it like this because it's not going to be sparse anymore, so it's going to take a long time. So I'm going to save it. Scale data. And then I'm going to watch it, see it like in the viewer. And you see that is not even full of dots. It has numbers for everything. So these are the sorts of modifications that we do to the data after, like through the analysis. And then the dimensionality reductions that are here, dimensional reductions, are the things that are used for plotting. So if you wanted to do, for example, something that comes up usually in the papers of this object, then you could use the dimplot function, and you can tell it use a particular reduction. I'm going to use the reduction that is the later one, the last one that I use. And then you can change the colors here with the calls. You can see do blue and red, and so on. So you can just explore through the options and see what you can do. And with that, I think that this is enough for Sarat for now. Again, explore, look at the tutorials, and if you have questions, let us know. Okay. Any questions so far? Yes.
I was wondering, what does the 0 cases that you should remove come from? Like that gene not expressed?
No. So the 0, like for example, cells that have a total count of 0.
Yeah, and feature 0. Right?
Yes.
So that example.
So that case, that's a beautiful question because it leads us directly to the next part. But long story short, those are empty droplets. So remember that this data set that we downloaded was from a 10x experiment, which is droplet based. So when you have that, the droplets can have ideally they have 1 cell, but sometimes they're empty. Sometimes they have more than 1 cell. So if they're empty, they don't have anything, and you result with 0, 0. Okay. So perfect question. Thank you so much. We need to do QC because sometimes we have empty droplets, or sometimes we have things that we don't want in our analysis. And to do that, you can get a head start by looking at the automated QC reports that are generated by the pipelines that are used for making the counts matrix. So Cell Ranger gives you a report that looks like this, and SplitPipe gives you a report that part of it looks like this. In the presentation, you can click here for links on how to read these reports in depth. But briefly, what I really want to look at is at the number of cells that the pipeline called. Like in this experiment, they got like almost 6,000 cells. And in this experiment, they got 200,000 cells. So is this within your expectations? Is this the number of cells that you wanted to capture or to sequence? And I personally also like to see at the mapping statistics because it's a gut check. If you have good mapping statistics, that means that one, you run the pipeline with the right reference. But if you have bad mapping statistics, it could be for a variety of reasons. Maybe you use the wrong reference, or maybe you have contamination, or maybe there was a problem with your sequencing, or who knows. So it's good for troubleshooting or just for gut checks. Anyway, this is the automatic quality control, but you also have to do your own. And basically what the goal is to filter out the low quality barcodes, either because they're empty or sometimes because they have information within, but it's just from sequences that were in the ambient. So they are just contaminated. And the general approach to do this is to look at the distribution of the values of different quality metrics. Normally, like we use the number of counts per barcode, and you can plot them. These are different examples on how to plot these metrics. This is called a cleave and toe plot, in which you order the barcodes by the number of counts of transcripts that you get. And then you observe the distribution. Or you can see use of violin plots. And in this case, this is an example of the percent of mitochondrial reads. It is widely accepted that barcodes that have a very, very high percent of mitochondrial reads might represent like dead cells. So you want to also look at that distribution. Or you can look at the total count of total unique genes that are found in a cell. And I like to use histograms because you can see that most of the barcodes have like around this number of cells. And then you have barcodes that are very few, that have very few genes. So after you observe this, then you can establish rejection thresholds. You can say, everything below this, I think is garbage. So it's not going to be part of the analysis. And everything above this mitochondrial percent, and so on. So this is an approach, but some people also start with things that they've looked, thresholds that they've seen used in the literature, and this is very valid. But I would caution people because whatever worked for some sample and some tissue and some technology doesn't necessarily work for yours. So honor the uniqueness and be curious of what your samples show. All right, after the QC, we go through the wrangling of the data from the raw counts to these decimals and these other decimals and so on. So this is the normalization step. This step is done so that different cells are comparable, and we try to account for technical and biological variations that are not the main interest of the experiment. So, for example, these plots were made with the geoaccession, with the data from the geoaccession. This shows the number of row counts that has each barcode. It's a histogram. And you see that it has this shape. But after I do normalization by depth, dividing each by 10,000 and take a log of that value, then this distribution changes the shape, and we see that there is some other thing going on here that wasn't visible here. So this is a common approach to better use the quantified hits in the counts matrix, but there is still discussion on what are the best practices to normalize the data. Some people think that normalizing by depth, even though it's great for technical, for getting rid of technical issues, might confound biology. So stay tuned, and even though normally we do this might change in the future. All right, next step is the dimensionality reduction. What is this? I've shown you in Scampari and Pandas how big these datasets can be. And the geo that we downloaded is just one sample, very like one thing. even with that, the matrix, the count matrix, had tens of thousands of cells and tens of thousands of genes, and many of them were zeros. So if you want to calculate things with that, that would make it very slow and you will need lots of resources. So what people do is just try to take that data and wrangle it to make simpler representations. Instead of taking tens of thousands of genes, they do a dimensionality reduction option or algorithm that's called PCA. And instead of the 10,000 of genes, then they have 50 principal components. This is a principal component analysis, and so on. So you can use different algorithms to reduce the dimensionality of this humongous matrix. There are different algorithms and they are optimized for different things. So in these plots, usually that come up in papers all the time, usually people are representing the cells, the humongous data sets, using only the first two dimensions of either reduction that they are using, either the UMAP or the PCA. But believe me, these plots are representing exactly the same data set. I made them myself yesterday. And they probably give you a very different intuitions of what's going on with the data. So my point here is that as with every representation that we do, these representations can only tell you part of the story. So if you look at the UMAP, you could conclude that you have two different cell types. One very different cluster that actually represents the stem epithelial cells. This was the annotation that we did later. And then you have these other things, right? So the UMAP could be used to say, hey, we have different clusters. But the wrong part is that you would maybe assume, hey, this is very different than the others. It turns out that with the same data, if you look at it with a different reduction, you see that maybe these ones, the stem epithelial, are not that different from the others, but they are actually in a pathway of differentiation. So please be aware of the conclusions that you try to jump to when you see one of these plots. They have their strengths. UMAPs can show you different clusters, but they are not really good at telling you how different things are. So instead of using UMAPs, let's try to use an alternative to see clusters. And we can use something that's called a clustering, which is this sort of graph. I'll explain it in a second. The goal of clustering is trying to group things that have a similar identity so that we have more robust signals before we do any comparison or try to answer any questions. But how similar something is a relative concept. You can say all humans are similar, or you can say some humans are a little different and some other humans are a little different. So you can modulate how similar something is. And we modulate this with a parameter that's called resolution. So let's go to the class tree. If you, a class tree shows you all your cells in your sample represented by dots or circles. And they are colored, they are grouped depending on the cluster that they were assigned. If you have low resolutions, you are very accepting. Everything is kind of similar. So you are not trying to go to the very nuanced differences. But if you increase the resolution, going down in this clustering tree, then you see that things that seem to be similar before actually divide. And you have some different, like two different subclusters from here. This process of increasing the resolution helps you differentiate things like that at different levels. You can say at this level, these cell types are something, and then you go down, and then you have something a little bit different within. Anyway, the point is that you want to balance how detailed you want to be which is increasing the resolution, with how open you are to noise. Because if you go too high in the resolution, then the clusters that you get might be driven by noise in the data instead of real differences. So what I like to see in my clusters is that the overall structure resembles what you're expecting to see in your cells, in your samples. For example, this graph was from a sample that we expected to find 4 different cell types. And you see that even from low resolution, there are 4 branches, and then you can increase the detail and you can say that, oh yeah, there are different things here, but overall we have 4. So that's a good gut check. Okay, so you have if you have different options to say to group your cells, then how do you know if you are grouping the cells correctly? And this is one of the main things that you have to decide when you're analyzing your data. There are, in my opinion, there is no one single good resolution. So you can choose different things depending on your questions, but there are definitely bad resolutions. So you want to select one that separates the cell types that you're interested in. And so that each cluster has enough cells so that the signal is robust and it's stable, it's not driven by noise. So try to get the resolution that checks these constraints, and then that would be the one that you would choose. And when you have chosen it, you have to identify them. What are these clusters? And maybe this is the most challenging part of the analysis. There are algorithms that are coming all the time trying to help with this, because we have to base our identification in what's already around. So either you know some marker genes for the cell types that you are interested in, and you can project the expression of those genes into a UMAP, for example, and see which clustering resolution separates this because it has high expression of your marker gene. That would be a way to go about it. Or you could use annotated atlases, so atlases or annotated references that have already gone through the annotation process. So some examples of these are the Cell Typist or even Cassia, which is a recent but seems to work well with some samples. And with that, I leave you with an annotated data set and with Sarah.
Thank you, Natalia. So for the last section of our tutorial, I will discuss what you can do with your annotated data set and how you can go about exploring biological questions. So earlier, Natalia showed this diagram showing that you have your annotated gene expression data and you can take several different analysis avenues. We've done many single cell projects in our group, and sometimes we do all of these different types of analysis for a given project. And this is not an exhaustive list, and the intention here is to give you an overview, an introduction. We're not going to get into heavy details here. So for differential expression analysis, many of you may be familiar with this from doing bulk RNA-seq. It's the same type of concept. We're looking at genes that are differentially expressed between our cell groups. This image here is from a study that I was co-author on, where you can actually see individual cells shown here and the expression levels across the different annotated groups shown here on the bar at the top. And then what you would do with your differential expression gene list is then you would look at pathway upregulation or downregulation. When we do differential expression analysis for single cell projects, we prefer to do pseudobulking if sample replicates are available, which we really strongly recommend. And so just a little bit about how pseudobulking works as a concept is essentially you're taking your single cell data and you are merging your your clusters per sample. And so in Seurat, we would use this aggregate expression function to be able to merge our gene counts for cells within a cluster. And essentially, you're calling that cluster of cells is like a sample. And so we have this example over here at the right where we have these different, I don't know if I think they're guinea pigs, getting either cheese or a diet of wheat. I think that's wheat. And then, so we have these replicates. This is our single cell data, and we pseudobulk it essentially. And so the annotation here that we're using is T1, T2, T3, those are the treatments. C1, C2, C3 are the controls. And then C0 is cluster 0. We haven't actually named these as a certain type of cells. We just have 3 main clusters, 0, 1, and 2. And so in this table, we would then have C1, T1, C1, T2, et cetera. And then you can use the well-established statistical methods for differential gene expression analysis, such as EdgeR and DE-seq2, to do your gene expression, differential expression testing. And it's really important to remember that you want to apply normalization and use covariate modeling just as you would do with a bulk RNA-seq experiment. So then another approach that people use, and this can also be commonly used when you don't necessarily have a clear treatment control condition, and you want to look at how gene groups might correlate with each other. You can use gene network analysis strategies. And so you can correlate those with your experimental conditions. There are some newer methods, such as this topific method here, where you can try to assign multiple gene programs to individual cells, where a cell might not just be doing one type of function, right? It's going to be doing multiple things. So you can say that it's a group of different cellular programs that are active in that cell. And so, a couple of the approaches that are available are matrix transformation approaches. So I've listed them out here, and then also unsupervised machine learning methods such as LDA. And then this space is also growing. There are more and more methods out there. And so this is definitely not exhaustive. And then another important tool we use for exploring our data is proportional analysis. So we end up getting different proportions of cell types within our samples. So this diagram is showing that here. And what we want to know is, are the differences, so between these F samples, are those really distinct in terms of this pink cell cluster versus the other conditions? And so we want to do some testing to know whether this is real biology or is it just a batch effect. And so a package that we use is the speckle-R package, and there's a function called propeller that is nice for doing this type of statistical analysis. It does require replicates, and there are multiple types of statistical tests that can be applied, including linear regression modeling or ANOVA or t-test approaches. And then another approach is what I'm broadly calling temporal modeling. And so the idea here is that you want to kind of think about how your single cell data set, how groups of cells might be changing. It could be something like looking at the development of B cells, or maybe there's a difference between experimental treatment where you apply a treatment and then your cells change. So this diagram here is showing HSCs, so stem cells differentiating into different lineages, and the pseudotime calculation for that. And so there are a variety of different software programs for doing this type of temporal modeling. There's pseudotime software programs, some of which I've listed here, and then also RNA velocity software. And so I want to just get into a little bit about what these two kind of main areas of analysis are. So with pseudotime, you're trying to infer time essentially from your single cell gene expression data. And there can be different structures that are possible. And most programs ideally expect the researcher to determine the origin or root cluster that you're going to use for the analysis. Although in practice, some of them will choose a cluster for you. So be careful about doing that. This paper here, I'm showing an image from this paper by Salins et al. In 2019, is showing a variety of different methods. Some of them can give you cycles in your data set, but other ones do not provide cycles. They'll just provide a linear pseudo-time plot. And it's important to remember when you're doing this analysis that you want to have a reason to do this analysis on your data. So you would expect that your cells are differentiating. If you just have a treatment and control where you have cells or you gave them a drug and they're not really expected to be having some sort of development response, then this type of analysis may not be appropriate. And again, this is not an exhaustive list of software. There are many other ones available. This table from this paper is much, much longer than what I'm showing here. And it's also from 2019, and so newer methods are now available. So then in addition to pseudotime, there's also RNA velocity analysis. And so what's going on here is you're actually comparing the relationship between unspliced pre-mRNAs and spliced mRNAs. And so this requires special analysis of your BAM files that you get from either Cell Ranger or SplitPipe. And so this example here from this study from 2018 is showing the different cell types that have been annotated, and then you can get these plots where you have these little arrows, these vectors essentially showing how the predicted cell states are changing. So then we've covered some of these ideas of how you could explore your data. Another thing that's important to talk about is that there are other types of assays beyond just gene expression, beyond RNA. And so there are kind of three major types that people are looking at, but I expect this may expand in the future. So ATAC-seq, single nucleus ATAC-seq, is a single nucleus assay example, and you're looking at open chromatin regions from your single nuclei. Currently, the commercial platforms do not provide any sample multiplexing options, so that's important to know. There are different QC considerations for ATAC-seq versus RNA-seq. And it's important to remember that you need to use an RNA-seq reference usually to annotate your ATAC-seq data, to annotate your cell clusters. And then multi-omic libraries are currently available from 10x, where you can measure both ATAC-seq and RNA or gene expression data from the same nuclei within a sample. And so there's limited multiplexing for this option, and the quality of nuclei is really crucial for success for this type of project. And then lastly, people often, particularly for immunology studies, are interested in surface receptors. So VDJ libraries are a way to look at that information. Those are types of library preparation kits are available from both 10x and Parse. Usually this is information that's provided along with a single-cell RNA-seq assay, so they're done in tandem. And then just like BCR and TCR sequences can be measured, you can also measure other surface proteins with a similar approach using oligo-DT tags, oligo tags. And an example is BioLegend for one company that provides those. And so this brings us to the close of our section, or of this section of the tutorial. I want to put up this this last slide to show you the template for single-cell experimental design, and also links for bioinformatics help and also tutorials. Our team is hosting an informatics interest group, a hybrid meeting every 2 weeks where we talk about different algorithms. And then, as Natalia noted earlier, down here in this section, we have a lot of different resources for you to use as you start exploring single cell experimental design and analysis. And we will be sending out an email with a link for this tutorial video and this webpage. and we will be requesting feedback from you. This is a initial demo version of the tutorial. We're planning on making it a little bit more interactive in the future. And with that, we can take any questions.